Okay. All right. So another major component, probably the second major component of cell membranes, are these things called proteins. Proteins, again, the structure is 100% what dictates how well they function or what they're able to do. Okay. So and what it comes down to when we look at the level of protein organization really is their final conformation or their shape. Okay. So the shape of this protein is going to dictate what that protein is able to do, how it's going to be dealt with in terms of other cells, and what functions that protein is going to be able to do. So we have two classes of proteins. We have proteins that are only found on one side of the membrane. Those are called peripheral proteins. And then we have proteins that run the whole length of the phospholipid bilayer, and those are called integral proteins. So hopefully everybody feels comfortable with the way I've laid this out, but it doesn't necessarily have to penetrate all the way to the middle. It could be found on the outside really to be a peripheral protein, but an integral protein has to completely go through both sides of that membrane. So it has to have an internal and an external side. And this is going to be really advantageous if we want to be able to ship a particle through the middle of that protein. So we can manufacture the protein, have a certain size channel in the middle, certain specs to that channel, so that only certain particles are going to be able to pass through it. And that's really the benefit of having these embedded proteins, is that it can be highly specific and allow the cell to function in a very specific way. Okay, so again, with all proteins, these R groups are going to play a huge role. The R groups are what really provide the variation in amino acid sequence from one amino acid to the next. And the R groups are going to dictate whether a protein is hydrophilic or hydrophobic, and that's going to dictate how it orients within the cell membrane and what it's able to do as part of that cell membrane. So just keep that in mind as we go through the rest of these examples. Okay, so here are proteins embedded in the cell membrane. Try to show you an integral one here, and this is a channel that would allow maybe a charged particle or maybe a specific polar molecule that's a little bit larger to come through that channel. Okay, and then these here are peripheral proteins. You notice that they don't go completely through the cell membrane, and they could be internal, like this one over here is more on the inside of the membrane, and this one's more external. Okay, so in terms of proteins, there's a lot of different things that these proteins can do. They can have enzymatic function, meaning they're going to break down or carry out reactions on the outside or the inside of the cell. One really important role that they have is for cell recognition. Think about how important it is for our bodies to be able to recognize our own body. If you have a white blood cell that is really a specialized killer and it just going around, it has a hard time recognizing yourself from a foreign entity, it would cause a lot of damage to your body, and in some cases it does. Okay, but that's all proteins, really, glycoproteins and things of that nature that allow our cells to recognize self from non-self, and also what target certain hormones and stuff are trying to reach. So when cells are talking to each other, these proteins are a huge part of it. Like you can have a hormone sent off from the pancreas that is going to interact with a cell of the small intestine, or any number of cells really. But in order for that interaction to occur, a protein is going to be involved on the surface of the cell. Obviously, transport, we're going to get into a lot of detail about this, but there are carrier proteins. There are proteins that are able to change shape, that are able to be modified to enable certain things to move into or out of cells. And this is really how we have specific control over what enters and what leaves, are these proteins that are going to be used for transport. And then the last thing that I really want you guys to feel comfortable with is proteins. They can be made in any orientation, really. There are perfect anchoring points for extracellular matrix or materials like microtubules and things to attach to, or different network of fibers, both on the outside and the inside. So cytoskeleton can anchor to these proteins, and it allows cells to connect to each other. So there's a lot that we can accomplish with these proteins, and really you have endless iterations. Proteins really can be assembled anyway to create any different function in life, and that's why they're so widely used. Okay, so another really big component of the cell membrane, these things that are lipid molecules called cholesterol, they're identified typically by a 4-ring structure. These are carbon rings. Got 3 hexagons and a pentagon there attached, and then we have these lipids are non-polar, of course. They're going to be embedded in the cell membrane like this. Okay, so they'll be found in between the phospholipids, and what these things are going to do here is they're going to allow the cell to kind of maintain its integrity in various conditions. Okay, so if the temperature is really hot, like in our bodies, you run at like a 98.6, which is like 37 degrees Celsius. These guys prevent the membrane from becoming too fluid. So they stabilize the membrane, and they allow it to kind of be maintained. Likewise, if it gets too cold, these things are gonna maintain some of the fluidity of the membrane, so they prevent it from really getting iced out and not being able to move, or to stay fluid is really the best way, I guess, to say that. So the last thing I wanted to get into with you guys is just a couple other more specific examples. Okay, so this is looking out at the outside of the cells, inside down here, and we have these combination molecules. Okay, so this is a protein that I depicted in blue, and attached to a protein we have several sugars. So this is a carbohydrate that's attached to a protein. So we call this molecule a glyco. That's the part referring to the carbohydrate, and then protein. Hopefully that makes a lot of sense for you guys. And then over here we have a series of sugars that are attached directly to the phospholipid. So that's called a glycolipid. Okay, and both of these are used for cellular identification. So like, I don't know if you've heard of this yet, but we'll talk about it later this year. We have A, B, O blood groups, AB blood groups. These things are really come down to these carbohydrates that are found on the outside of the cell, and that's how your white blood cells are able to distinguish what type of blood you have and know not to attack it. So they're vital in terms of our ability to really establish cell-to-cell recognition. Okay, and so hopefully everybody makes sense of these words. Again, glyco is in reference to the carbohydrate aspect of the molecule, and then the second part is I hope pretty clear. Protein, if it's a glycoprotein, is half carbohydrate, half protein. Glycolipid is going to be half.
I found this on the web for haste.
Half carbohydrate and half lipid. Okay, so the cell membrane is going to be comprised of phospholipids, proteins, cholesterols, glycolipids, and glycoproteins. And at this point, I hope everybody knows that this is continuously changing. It has the ability to be altered based on function. That the components are going to be completely plug and play. They vary throughout the cell's lifetime, and they vary cell to cell. This collectively we consider in biology to be the fluid mosaic model. The fluid aspect of this term means that it's constantly changing, and the mosaic means that it has various pieces, which we described up here above. And I hope everybody feels comfortable with everything we try to lay out here. If you have any questions, make sure that you ask. All right, have a good day.